Welcome to our next session on our real-world impact and regulatory decision-making track, where we are seeing a lot of good examples on use of modeling simulation and tools for the development of drugs and other real possibilities in the pharmaceutical scenario. So here we have this session where we're going to have two presenters, which is the first one, Maxime Merdy, which is Associate Director on Research and Collaborations on Simulation Plus, and Jonathan Chauvin, which is President of Lixoft Division, also Simulation Plus. And the presentation is going to be GastroPlus simulations to Monolix for trial design. So please, Maxime, Jonathan, the floor is yours.
Thank you, Gustavo, and thank you everyone for joining today. So the goal of this presentation is to show some sort of a case study on how we can use multiple platforms and how we can combine PPK and population PK to support the development of a generic drug product, all the way from the concept of the drug all the way to the trial design. So the objectives of these case studies were the following. The first one, it was to validate a mechanistic in vitro to in vivo correlation, and I will refer to IVIVC as I speak for the next slides. Then we wanted to predict the clinical PK of the formulation variants, perform a few virtual bioequivalence assay within GastroPlus or using the PPK model approach, and then really switch from PPK to PopPK to design a bioequivalence clinical trial. To obtain these objectives, this is the workflow we have conceptualized with Jonathan. The first one was, the first task was we started with in vitro release data for multiple formulation variants for whom we had the corresponding clinical PK profiles. With those data, we used GastroPlus to develop an IVIVC. Using this IVIVC, fully validated, we could simulate clinical PK formulation variants and then perform PVBM PPK-based VB. I would like to refer to Rodrigo's presentation that kind of integrated some of those concepts. Then, because those platforms between PPK and PopPK are connected, we could simulate populations that were ready to be read directly into the software Monolix. And this is where population PK comes into the game, where we could first build a reference model in Monolix, create and understand the impact of formulations as a covariate, and finally, using the Monolix suite and more specifically Simulix and PK Analytics, design the bioequivalence trials. So let's first have a look at what we've done in GastroPlus and the PPBM PPK part of this project. Just before I jumped into the results in a way, a quick reminder of what is an IVIVC and what is the definition of an IVIVC by the FDA. An IVIVC is a predictive mathematical treatment describing a relationship between an in vitro property of a dosage form and the relevant in vivo response. So long story short, for what it means for us, the IVIVC is a development of a mathematical relationship that we have here between an in vitro release profile and an in vivo plasma concentration time course. How do I need to adjust my in vitro release profile mathematically so I can predict the in vivo behavior of a formulation? The drug we're going to talk about today in this case study is valproate. And it was because for this drug product, we have data coming from a paper published in 2005, where we have all of the in vitro release profile for multiple variants and the corresponding in vivo PK profile. So using those first clinical data, what we've done is using the IVIVC modules in GastroPlus, we have deconvoluted what will be the in vivo release profile. And you can see here on the screen in the middle, in red, what is the in vivo release profile for the slow formulation, in blue for the moderate, and in green, for the fast, and what are the corresponding simulations for those formulations if we use those in vivo release profile. Once we had those in vivo release profile, and because we had the in vitro release profile, we could build the IVIVC. And you can see the equation here, which is a polynomial of second order. On the x-axis, we have the in vitro release, and on the y-axis, we have the in vivo release. So we were able to build a level A IVIVC. Using this relationship and having this relationship, we had to validate it. Following the FDA guidances, we first did an internal validation, and you could see that all of the individual criteria are below 15% for both the Cmax and the AUCs. So that was validated. And the mean absolute percentage protein error were below 10% also for the Cmax and the AUCs, meaning the internal validation was validated, was accepted. For the external validation, we used data that were not used to build the IVIVC. And for those formulations, the criteria are simply the mean absolute percent predictive error that has to be less than 10%. And it was the case for both Cmax and AUCs. So once again, our IVIVCs was able to be validated also for the external part. So we are fairly happy at this stage, and we had a full IVIVC validated. The next important step was to verify that the model is able to predict the inter-subject variability, because we want to design a clinical trial, so we want to make sure the population generated by the BPK model can reproduce what is observed in clinic. So on the screen here, you can see the 25 simulations for 25 virtual individuals that were representative of the clinical trial demographic. And you can see that the spread of the simulation is fairly well aligned with the mean data point in red and the standard deviations at all time points. So we had a model that was able to fairly well represent the inter-subject variability. So having that checkpoint validated, we could then move into designing the formulation variants, making the simulation, and generating the population. We kept it simple at this point, and we only simulated or created 2 formulation variants that had a release profile that was 10% and 20% slower than our reference drug product in blue. Using the PBM-PPK model, we could predict what would be the PK outcome in a representative individual, and this is what we see on the right. And to get the simulation, we use the IVIVC that we have just validated. Of course, representative individual, it's not enough to assess the bioequivalence of drug product virtually. So we actually simulated 3 populations, one population that received the 3 formulation variants, and we created a population of 100 individuals, and we integrated the intra-subject variability. Actually, the way we did those simulations was to simulate 2 crossover. Once the population received the ref, and then what we call the minus 10% variant, and the other crossover was the reft, follow was the minus 20% variant. We went a step further, and we compared those populations directly into GastroPlus. The minus 10% formulation was found to be bioequivalent to the reference in the software. And the minus 20%, although closest to the BE limits, was also found to be bioequivalent. So at this stage, we had formulation variants that could be interesting to move to the clinic, but to go a step further, we wanted to investigate really how to use those data and how to be able to perform a clinical trial. So what we've done is we use a new functionality in GastroPlus, which is to create a population in a specific format, like a specific layout in Excel, that can be read directly by the Monolix software or population software. And this is where population takes the lead, and I will let Jonathan present the rest of the slides.
Thanks a lot. So if we can go on the next slide. So the goal of the population is to be there to go to the next phase. So with GastroPlus, we have a very nice answer with this formulation. What's from a theoretical point of view and our modeling point of view, is it bioequivalent? Is it sufficient? And we have this hypothesis. Now the goal is to go toward the clinical trial and to say, okay, we have this theoretical or model answer. How do we go from that to a real clinical trial? How do we design that? Toward that, we're going to have several steps. First of all, we're going to go from this perfect concentration defined by GastroPlus and to have measurement noise representative of what would be closer to the reality. We develop a population model to be able to easily simulate clinical trial. And in that case, clinical trial means a lot of different things. It means how many individuals do you have in each group? Do you do only crossover? Do you want to do parallel? Do you want to have repeated crossover? So you have a lot of different possibilities. What could be the impact of a specific covariate? So we can have sex, we can have age, we can have weight, things like that. Are there separate group of things that we should consider in a specific formulation, or do we see specific population within what we have simulated that are more closer to the bioequivalence? And with this population modeling, using the biological model of how the drug is going into the body and how it reacts with the patient, the impact of the covariate on this variability, we are able, and also the formulation, we are able to simulate this trial and look on specific trial that we design. Is it bioequivalent? If we were doing that in reality, and what's the probability of success of this kind of trial? So that's the global framework of the population approach, going from the output of GastroPlus. We believe that this formulation makes sense toward a real clinical trial. So thanks. So the first thing is to really add measurement nodes. So that's what we've seen is that the concentration measurement proposed by Maxime were more or less perfect and very dense. We had a lot of time points, and two, again, kind of clean compared to what is reality. So we have the possibility to add measurement noise and modeling noise that we can grab from publication of typical values. And what we see on the left is the real value coming from GastroPlus. And we are adding a lot of noise and measurement error on it, on the right. What we see is that the mean curve is basically unchanged, which is quite normal. We have much more variability on the measurement. However, we remain with the same trend, and we should have no specific impact by adding that to the bioequivalence. If we go to the next slide, what we see is that it will increase the variability of the measurement, but has no impact on the several mean PK metrics, nor on the bioequivalence results. If we look on the left, we have the bioequivalence results with the original data set. So on the left is the reference minus 20%, and on the right minus 10% on 4 PK metrics. So typical AUC, Cmax, or the AUC between 0 and 24. And if we look at what's with the noisy and more real data set on the right, we see that we do not have exactly the same value, but we have very close value corresponding to the fact that adding this noise on the real trial does not impact the result that we're gonna have. So this first step to go closer to reality makes sense for that. If we go the next step to be able to model that, so what we're going to use is to use Monolix for population PK modeling, starting on the reference formulation. So what we see on the plot is the concentration with respect to time, and we see the yellow dots are what we see in the concentration, all the concentration. What we see in white is the mean value and the standard deviation on each time point. What we see on the absorption phase, that there are two kinds of absorption. The first one really between 0 and 5, and another one, so really two shapes. And what we see is that really we have a double absorption model, and we have all of these models are already covered in Monolix library. So we'll be quite easily able to model that. And what we model is two things, is typically model the mean population, but also the variability. So we're gonna, if we go to the next slide, sorry. What we'll be able is to have a PK model with a double absorption. So first of all, a first order absorption with a lag time and a zero order absorption with a longer lag time. And what we see is that what we want to catch is this biological behavior, and also to see the variability, so to be able to estimate on this light tide, this volume discriminants, the variability among the population. To be able to validate that, so what we see that is really in a couple of clicks in Monolix, we're able to look at diagnostic plots. So everything is embedded in the software to make the user focus on its work and not on how to make it work. And two classical diagnostic plots for that. On the left, the observation versus prediction in the log-log scale. So showing that if we are at lower concentration or high concentration, we are close to the estimation that we believe that are the good one. And on the right side is the virtual predictive share. So as we have a population model, we're able to resimulate a sample of it, and we see that our model predicts well with this double absorption, really what we want to do with the behavior, the global behavior. So global behavior, meaning both the biological model representing the biology of the drug, but also the variability within the population. If we go to the next slide, the next step is to go with that to include the variation of the formulation in that. So we can use the formulation as a covariate and also to include biovariability to look at the formulation. And this covariate and all the covariates are impacting not only the bioavailability, which is obviously the first parameter that is impacted, but also we can see the different repartition between the different absorption, and also maybe the lag time and things like that. So the population is also going to say what is the impact of the formulation, not only 10 and 20, but on a linear scale on all the kind of formulation we can have between those. So the real value of the population is to really be able to describe. And what we're going to do next is to be able to simulate clinical trial based on the information that we have. So the population model is really there to make sure we understand both the biology and both the statistics of the population and the formulation. If we can go on the next slide. So why are we doing that? Because I mean, that's nice curves, nice layout in Monolix, but there is a goal behind that. So it's to really answer questions. So the first is to use this model to perform simulations to answer operational questions. So the first one that we're going to ask is, how many people should I enroll in my clinical trial to ensure that my trial shows bioequivalence? If I have 2 individuals, I have guaranteed that there is no bioequivalence, but that's going to be very cheap. If I put 1,000, that's going to be very expensive, and we're going to be sure that it's going to be. Where is the trade-off between that and the impact of the formulation is something that we'll be able to see. Again, when we're going to make blood measurement, how many concentration measurements should we have to ensure bioequivalence? Should I make 1, 10, 20, 1,000 measurements to make sure that we can have that? And finally, what we can design the bioequivalence trial, but also what's the impact of the formulation if we are looking between two formulation or also what are the variation of the parameters that make sure that we're still bioequivalent. So in the other way around, how this information could impact also the design of the formulation. In all these cases, what we do is that we are using simulations to generate simulated data set, and we use PK analytics to perform the NCA and the bioequivalence, answering that question. So for the first question, what we're asking is how many individuals should we have to ensure that my trial shows bioequivalence? So what we do is that we simulate a trial. So in that case, crossover, but we can simulate any kind of trial with that, with an individual per group, and we compute the bioequivalence on this trial. And as we are just making a sample, so we tried 10, 12, 20, 100, we are just making draws of a specific number. If we draw another 20 people of the same trial, that's not going to be the same individual. So what we want to see is what's the variability or the uncertainty associated to that. So what we see is that if we are doing with the 10% variation in the formulation, we only need 15 persons to ensure bioequivalence. That's what exactly Maxime showed, that it was very closer to the reference formulation. So as we were quite less far from the boundary, we do not need a lot of individual to make sure that it's going to be fine. On the opposite, with the 20% difference formulation, what we've seen is that we were still okay from a theoretical point of view, but we were closer to the boundary. And in that case, if we put 25%, we get just 50, 70% of probability of success of my trial. So that's going to be very risky. So I need at least 75% to have a good probability of success of my clinical trial. So in that case, it just shows that maybe that the 20% formulation is better from a lot of different point of view. But if I would like to use this formulation to go to the clinical trial, then it's going to be much riskier, or I need more people, so a more expensive trial to ensure that this formulation is going to be bioequivalent for that. So that's the first answer that we have with simulation. The other one is that we are making a lot of measurements, concentration measurements, and we can say this is a typical pattern of the times that we can take blood concentration. And again, that's very costly and time consuming to have all this measurement. What's the impact of removing one time so we can test a lot of different ones? Again, we simulate different populations. We compute bioequivalence on this trial, and we use replicates to limit the uncertainty to compute the power of the trial. What we've done is with the 10% concentration with 20 individuals, we removed the last two one, and took a different final one. And what we've seen is that there is no impact at all on the bioequivalence study, meaning that in that case, we can not make the last 4 or 5 measurements, and it has no impact. So it has a lot of reflections to say, okay, we can shorten the trial and make it shorter in terms of time and less costly in terms of measurement. And finally, the last question that we can ask, the next slide, is to say, how can the impact of formulation of changing the fraction? So if I know that my formulation can also impact the fraction, so the fraction between the two absorption phases, what's the limits to make sure that we have this bioequivalence? So what we've done is to simulate the population model varying this fraction between basically the two absorption, where we believe that we can have an impact. And what we've seen is that we can change it a lot. So it was at the reference as closer to 0.9. And if we go over, or if we reduce it to 0.75, we still ensure the bioequivalence. So that's also answering the other way around. I can change formulation to have an impact of these parameters by 25%, and we see that it has no impact on the bioequivalence. So we can also go the other way around or use the population to inform what kind of formulation I can do to make sure that we're going to be. Finally, as a conclusion, we were able to validate using GastroPlus all the mechanistic in vitro in vivo correlation and predict PK of the formulations. And by that, with this reference one, we are able to go to the population modeling to go toward clinical trial definition, trial, and so the real question from an operational point of view, which has a huge impact on the cost, on the probability of success of the trial. And everything was done using the full simulations plus suite of application interoperable between GastroPlus and the Monolix suite and between the different applications of the Monolix suite. Thanks a lot.
Thank you very much. Thank you, Jonathan. Thank you, Maxime. So we have time for one question. So I'm gonna ask you, and it's from Mohammed regarding special populations design PK trials. So he's asking how Monolix suite will help the trial design a PK study in laboring and lactating women. So any comments on this?
Again, what we are doing is if we are grabbing the information, so typically if I grab the information from Maxime to have what's the impact of this specific population, we're able to integrate it. We have no biological knowledge of the impact of the covariate. We discuss and we can say, okay, it can impact this and these parameters. However, what we see there on the population framework is, does that make sense from a statistical point of view? So from how the biology is impacting that, I'm asking Maxime to say, okay, how do I integrate that? But how do we do that on a specific statistical way? It has to be impactful, and we can have this impact on covariates, such as volumes or such as absorption rate and stuff like that. But we are answering that from a statistical point of view, not a biological or mechanical point of view.
Yeah, and if I just may add on that, I think the best approach to answer the question from Mohammed is actually to use PPK model, because we can switch the physiology and go from healthy to special population, and it goes like further than just pregnancy. It could be liver impairment, renal impairment, pediatric population, elderly population, difference between male and female. So what we could do is, using the same approach as we have presented with Jonathan, but simulate different population that has different physiologies, either healthy or diseased populations, and then use Monolix to further evaluate if we need like specific clinical trial for those subpopulations, on top of the answers we would obtain just with PPK model.
That's great, thank you. So we have two minutes yet. So another question that came here. Normally distributed noise added in Monolix to individual plasma profiles from GPlus.
Yes, so we are using typical normally distributed noise on that. However, we can add correlation too between this noise, and the variance of this noise can also depend on the prediction. So basically, if you have a very high concentration, typically you have a higher noise than a lower concentration. So we have the possibility from a simulation or an estimation point of view to have different kind of structure, and that's part of the global statistical framework to say how do we understand the measurement noise from a data set. On this case, what we were to be able to add some representative noise. So either we can look at publication on this specific drug on how the measurement kit or stuff like that are impacting the modeling part, or we can have kind of standard rules that we apply there just to have something representative as a test case, as Maxime explained, just to present a framework on what we can do, just to say that we are not looking at a perfect trial and have a perfect response, but at simulations first, we try to focus on making that real.